Welcome back to yet another class of molecular basis of inheritance. Today we talk about the main principle that runs our life, that is known as, in molecular language, it is known as central dogma of life. Central meaning main, dogma meaning principle. So what is that which runs our life? What is that main principle on which our life goes ahead? See, the cell has the nucleus within. If I am talking about a eukaryotic cell, within the nucleus there is the genetic material. We call it genome. And this is being highlighted here. All these chromosomes shown here has been highlighted here. And one of the chromosome you see, which is a highly compact DNA. The DNA being opened up, and you can see the two strands, even exposing the bases. The complementary bases are being exposed. So each segment of this DNA on one strand is a gene. That means the sequence of bases on one strand is a gene coding for a peptide. That means a protein. So there can be gene sequences at different levels, at different regions coding for different proteins. All of these proteins are essential to run our life. They may be acting alone or they may be acting as a complex to carry out many cellular functions. So let's see how the whole information from gene is carried out in the formation of protein. How does the DNA express itself to make that protein? DNA cannot do it on its own. It needs a help. It needs the help of RNAs, several RNAs. At this level, we will be talking about mRNAs, messenger RNAs. As we know, information for life is encoded in the sequence of 4 bases of DNA. That is adenine, guanine, cytosine and thymine. The nucleotides with these bases, they are the language of DNA. So this sequence of bases directs the assembly of amino acids. Which amino acid should be first, which second, which one third, like that. A protein's amino acid sequence is directed by the sequences of those bases on the DNA. And another important thing, when we talk about eukaryotes, DNA is enclosed within the nucleus. Suppose this is the nucleus of the cell, and DNA being inside the nucleus, and protein synthesis factory, that is the ribosomes. Endoplasmic reticulum has ribosomes on its membrane, and the ribosomes in the cytoplasm. They are situated in the cytoplasm. So when DNA is in the nucleus, how does it instruct the protein synthesizing machinery, that is the ribosome, the sequence of amino acids that it should bring up? DNA being the boss should not leave the cabin. The cabin is the nucleus. Because in the cytoplasm there may be so many factors that can degrade DNA. The boss will lose its identity if it goes on every time coding for the protein. If it leaves the nucleus and goes to the cytoplasm to make up the protein, it cannot do that. It has to be highly secure. The information on DNA, that is the blueprint of life, should be highly secure. So there should be some messenger which would carry the instructions on DNA and get the work done, that can dictate life. So it remains there and it makes copies of it. That is called a transcript. That means when DNA never meets ribosome, we have to know how does it direct the protein synthesis. Sequences of DNA is copied to mRNA. This mRNA is the messenger RNA, or we call it a transcript. Also a nucleic acid. mRNA is also a nucleic acid, because if DNA's language is the 4 bases, the messenger also should have the same language of nitrogenous bases, except for uracil in place of thymine. So mRNA carries that instruction to the cytoplasm. So just see, this is a small strand of DNA being highlighted here. The sequence written here is just this particular segment on one strand. Remember, a gene is a segment of just one strand of DNA. When you take the other strand, the sequence written on the other strand is coding for a different protein. Likewise, the single strand also, at different areas there are different genes. There can also be sequences which are not coding for a protein. So non-coding sequences also could be there. So here I have just highlighted a small segment of DNA here in the blue. So you can see randomly I have taken a sequence. So that is one segment called a gene. Now this has got the instruction for a protein, for a particular protein suppose. So how will this instruction be carried out to the ribosome? This has to be transcribed into mRNA. This is the sequence of bases on mRNA. See, wherever thymine should have come opposite adenine, it is uracil because it is an RNA. So there is no thymine here. Otherwise this is exactly a complement of this segment. So an mRNA should be formed. That process is called transcription. We will read further. So when this instruction is copied to mRNA, the mRNA moves to the cytoplasm and the messenger RNA molecule will translate the language of DNA into protein. Protein's language is amino acid. Okay, so the mediator, that is the messenger RNA, knows how to decode that language into protein. Of course, there are other RNAs used for this process. Now, coming to a little more explanation of central dogma of life. See, this is the double-stranded DNA, and see the transcription makes an mRNA of one strand. You can see here thymine, so complementary is adenine, cytosine, complementary guanine, adenine. Now come to this area, thymine, here thymine complementing adenine. So the mRNA formed is complement to one of the strand. You can see the basis. Questions like DNA double strand would be given and you may be asked to write the mRNA formed from one of the strands of DNA that is asked in the question. So you should know how exactly the sequence would go. Now this strand which has made mRNA is complementary. The bases here are complementary to that strand, but the instruction is for the gene on the other strand. That means AG, AT except for thymine uracil. This is identical to the other strand. That means this is going to synthesize protein coded by the other strand. That strand is called the coding strand. We will go in detail when we talk about transcription in the next video. Then this mRNA has to move out. This is in nucleus. This is formed in the nucleus in eukaryotes. This has to move out of the nucleus to the cytoplasm and instruct the ribosome. You can see the picture here. Through the nuclear pore it comes out. It gets attached to the ribosome and on the ribosome it will allow tRNAs, transfer RNAs to decode the three letter bases which instruct each amino acid. So tRNAs also have a role. We will talk about tRNAs later. Transfer RNAs which are going to bring amino acids and assemble it one by one, forming peptide bonds in between each consecutive amino acids and make the protein, complete protein. So that happens in the cytoplasm. So messenger RNA acts as a carrier of genetic information from its site of synthesis. Site of synthesis of mRNA is nucleus in eukaryotes, and site of protein synthesis, it carries it to the cytoplasm, which is the site of protein synthesis, that is to the ribosomes. Another important thing, when we talk about central dogma, you should not forget that DNA making more copies of DNA, that is what we call DNA to DNA replication, which we have covered in the previous videos. That is also part of central dogma. Main principle of life has to make more and more copies of the information to be passed on to each cell. That is there. But what is the role? Why is this DNA being formed in every cell? So that the cell carries out its cellular functions, for which the instruction on the DNA has to be expressed as protein. So central dogma includes DNA to DNA replication. then DNA to mRNA transcription, and mRNA to protein, that is translation, translating the language of DNA to amino acids. Of course, after the chains are formed, peptide chains are formed, proteins undergo foldings, and that is how they carry out their functions. So we'll talk about the prokaryotes now. Prokaryotes do not have a nuclear membrane. So the mRNA comes immediately in contact with cytoplasm. This is a prokaryotic cell, and this is the genetic material which is somewhere in the cytoplasm in a region called nucleoid. So the strands separate for transcription. mRNA is formed by the enzyme RNA polymerase, DNA dependent RNA polymerase, because it is depending on DNA strand as a template to add mRNA nucleotides. So the mRNA is formed, and you see simultaneously it is being attached to the ribosomes and the peptide chains are formed. So this is a simultaneous process here. And the question like why DNA can, whether eukaryote or prokaryote, the question is why DNA cannot directly synthesize protein. As I already discussed, it will lose its identity every time it moves into the cytoplasm in case of eukaryotes especially, and even in prokaryotes. It has its own way of securing information. There is a high risk of loss of genetic material if it goes on carrying out the other work too. Enzymes and the pressure, osmotic pressure of the cytoplasm can disrupt the DNA. DNA being tightly packed, unwinding every time a protein has to be formed can be a very energy consuming process. It will not be energy efficient if it goes on doing that, because various segments have various genes, and every time carrying out this function can be a tedious job. So mRNAs, which are formed and degraded easily, they can carry out this work. They are meant for information transformation and carrying out protein synthesis, whereas DNA is for securing, keeping the information safe. So to keep DNA protected and to maintain its compaction, the cellular machinery uses mRNA to act as an intermediate. Terms related to this, again the same thing. What is transcription? DNA to mRNA. Flow of information in the cell is normally DNA to mRNA. mRNA makes protein. So uses of mRNA, just like why DNA is not used for translation. mRNA allows the cell to separate information storage, that is DNA takes care of that, from information utilization. mRNA takes care of that. It also gives the cell a greater opportunity to control the level and timing of expression of various genes. So mRNA can take care of as and when needed that protein would be formed, and DNA need not be carried out as a wasteful process of expression. Translation, mRNA moves to the cytoplasm, serves as a template for synthesis of protein, that is the language of amino acid. One difference here, when we talk about retrovirus, retro meaning back, reverse. So viruses, retroviruses are those viruses having genetic material RNA, not DNA. So there what happens is RNA is converted first into single-stranded DNA. RNAs of the viruses have to be delivered into the host cell in which they survive, in which they propagate, because they don't have their cellular machinery, so they use host cellular machinery. But host is having DNA. Whichever host they infect is having DNA. So how will it function? So they have to first convert their RNA into DNA, which is done by a process called reverse transcription. The enzyme reverse transcriptase is used to convert RNA into DNA first. Okay. Then that single-stranded DNA will make double-stranded DNA by process of replication. Then the same process will go on. That means again RNA would be formed and protein would be synthesized. This happens in retroviruses and it is known as modified central dogma. So that's all about the chief principle of life. What is transcription, how is it done, and what is translation we will take in the next videos. Thank you so much for watching. Good reading. Thank you.